bile secretion. Describe the composition, mechanism of secretion, functions and regulation of bile secretion. Bile is secreted from the liver and through the bile ductules it is excreted. Around 0.5 to 1 liter of bile per day is secreted by the human liver. For digestion and absorption of fat, bile secretion. Describe the composition, mechanism of secretion, functions and regulation of bile secretion. Bile is secreted from the liver and through the bile ductules it is excreted. Around 0.5 to 1 liter of bile per day is secreted by the human liver. For digestion and absorption of fat, the secretion of bile is essential. The cystic duct of the gallbladder forms a typical bile duct in conjunction with hepatic duct. The typical bile duct joins with the pancreatic duct and opens through the sphincter of Oddi into the second part of the duodenum. Composition of bile. Bile is a greenish-yellow fluid that is produced in the liver and stored in the gallbladder. It consists of water 98% and solids containing primarily bile salts and pigments and various ions. Composition of liver bile. Daily secretion: 500 to 1000 ml transparent alkaline fluid, light golden yellow in color, pH 7.8 to 8.6, isosmotic with plasma, water 97%, bile salts 0.7%, 120 to 180 milligram per deciliter. These are sodium and potassium salts of bile acids. Bile pigments 0.2%. These include biliverdin and bilirubin and its derivatives. Lecithin, a phospholipid 0.1%, 140 to 810 milligram per deciliter. It is present in inactive form when converted to lysolecithin active. It damages the cell membrane. Fats 0.1%. Fatty acids 0.15%. Cholesterol 0.06%, 60 to 170 milligram per deciliter. Enzyme alkaline phosphatase. It helps in converting organic phosphate to free phosphate. Electrolytes 0.7%. Bile acids. There are two types of bile acids, primary and secondary. The primary bile acids are cholic acid and chenodeoxycholic acid. In hepatocytes, the primary bile acids are formed from cholesterol. Secondary bile acids are deoxycholic acid and lithocholic acid. Secondary bile acids are formed in the intestine, where primary bile acids are converted into secondary bile acids by intestinal bacteria. Most of the secondary bile acids are excreted in the feces and only small amount is reabsorbed, so feces contain only secondary bile acids. Bile salts. Bile salts are sodium and potassium salts of bile acids. Bile acids are conjugated with taurine or glycine to form taurocholic acid or glycocholic acid. These acids then combine with sodium and potassium salts to form sodium taurocholate, sodium glycocholate, potassium taurocholate, and potassium glycocholate respectively. Bile pigments. Bile pigments are the excretory products in bile. Bilirubin and biliverdin are the two bile pigments, and bilirubin is the major bile pigment in human beings. Functions of bile. Digestive function. Bile helps in digestion of fat by emulsifying fat. Absorptive function. Bile salts present in bile helps in absorption of fats and fat-soluble vitamins. Excretory function. Bile pigments are the major excretory products of bile. Other excretory products include heavy metals, copper and iron, some toxins, some bacteria, cholesterol, lecithin, and alkaline phosphatase. Lubricative function. Mucin of bile lubricate the chyme in intestine. Bile stimulate the secretion of the hormone secretin. Bile maintains a balance between bile pigments and thus maintains a homogeneous environment. Anti-putrefactive. In absence of bile, putrefaction happens in large intestine. Laxative. It stimulates peristalsis, so acts as a bowel mover. Antiseptic action. It is the natural detergent of intestinal lumen and inhibits the growth of bacteria. Choleretic action. It helps secretion of bile by hepatocytes. Cholagogue function. Bile salts present in the bile causes the secretion of hormone cholecystokinin, which increases the contraction of gallbladder and thus increases bile secretion. Maintenance of pH. Because of alkali content, it neutralizes the gastric juice and also chyme, thus maintains optimum pH for digestive enzymes. Prevents gallstone formation. Bile salts keep the cholesterol and lecithin in solution and thus prevents the formation of gallstones. Give brown color to the stools. Mechanism of bile secretion. The secretion of bile consists of canalicular flow and ductular secretion. Bile secretion can be considered to occur in two stages: bile acid dependent fraction and bile acid independent fraction. Bile acid dependent fraction. Hepatocytes secrete bile into canaliculi, from which it flows into bile ducts. This comprises of large quantities of bile acids, cholesterol, and other organic molecules which are secreted by active mechanisms as micelles. This secretion is dependent of amount of bile acid absorbed. Bile acid independent fraction. As bile flows through the bile ducts, it is modified by addition of a watery, bicarbonate-rich secretion from ductile epithelial cell secretin, which acts on the ducts and increases the secretion without increasing bile acid. The gallbladder stores and concentrates bile. Bile is concentrated fivefold in the gallbladder by absorption of water. Enterohepatic circulation. Normal rate of bile synthesis is 0.2 to 0.4 gram per day, and almost 6 to 8 gram per day of bile salts are required to digest and absorb a normal meal. So enterohepatic circulation is necessary because of limited pool of bile salts available to help to break down and absorb fat. Enterohepatic circulation is a continuous circulation of bile salts from liver to bile and their passage into the duodenum, their absorption in the ileum, and then return to the liver as a mixture of bile acids and salts. 95% of bile salts are reabsorbed by the small intestine via the portal circulation to the hepatocytes and is resecreted into the bile, whereas 5% of bile salts are lost in feces 500 to 600 mg per day. Clinical implications of enterohepatic circulation. Because bile salts are required for proper digestion and absorption of fats, any condition that disrupts the enterohepatic circulation, ileal resection, or small intestine diseases such as sprue or Crohn's disease leads to decreased bile acid pool and malabsorption of fat and fat-soluble vitamins. The clinical manifestations of such diseases are steatorrhea and nutritional deficiency. An increase in fecal loss of bile salts results in watery diarrhea, since bile salts inhibits water and sodium reabsorption in the colon. Regulation of bile. The regulation of bile juice released into the duodenum after the meals is performed at two levels: regulation of biliary secretion and regulation of the release of bile from the gallbladder. Regulation of biliary secretion. The secretion of aqueous component, that is water and electrolytes, and the bile, do occur together but is regulated separately by the following mechanisms: regulation of bile-independent fraction of biliary secretion and regulation of bile-dependent fraction of biliary secretion. Regulation of bile-independent fraction of biliary secretion. The bile-independent fraction of biliary secretion refers to the amount of fluid containing water and electrolytes. Secretion of this fraction of the bile juice is controlled by secretin and vagal stimulation. Secretin, it acts on the ductal cells of hepatic ducts via cyclic AMP, the second messenger, and produces a large amount of watery fluid with a high concentration of bicarbonates. Vasovagal reflex, initiated during the intestinal phase of digestion, also affects the ductal secretion by potentiating the effects of secretion through the acetylcholine. Regulation of the bile-dependent fraction of biliary secretion. The bile-dependent fraction of biliary secretion refers to the number of bile salts secreted by the liver. It depends upon the following factors. The amount of bile salts secreted by the hepatocytes is directly proportional to the number of bile salts reabsorbed by them from the portal circulation. As the bile salts are recycled in the enterohepatic circulation, they maintain a high level of bile secretion during the digestive period. Bile salts and bile acids are the major agents that enhance the synthesis of bile. Secretion of bile from the liver and release of bile from the gallbladder is influenced by some chemical factors which are categorized into three groups: choleretics, cholagogue, hydrocholeretic agents. Choleretics: substances that increase the secretion of bile from the liver are known as choleretic. Effective choleretic agents are acetylcholine, secretin, acid chyme in intestine, bile salts, gastrin. Cholagogues. Cholagogue is an agent that increases the release of bile into the intestine by contracting the gallbladder. Common cholagogues are bile salts, calcium, fatty acids, amino acids, inorganic acids. All these substances stimulate the secretion of cholecystokinin, which in turn causes contraction of the gallbladder and flow of bile into the intestine. Hydrocholeretic agents. The hydrocholeretic agent is a substance that causes the secretion of bile from the liver with a large amount of water and fewer amount of solids. Hydrochloric acid is a hydrocholeretic agent. Regulation of the release of bile from gallbladder. The filling of the gallbladder by the bile is simply controlled by the pressure gradient. During the inner digestive period, the sphincter of Oddi remains closed. As bile is secreted continuously, it gets accumulated in the common bile duct. When the pressure of bile in the common bile duct rises, it forces its way through the cystic duct into the gallbladder. During interdigestive period, the pressure in common bile duct and gallbladder reaches 7 cm of water. Emptying of gallbladder. When the chyme enters the duodenum, the gallbladder is contracted along with the relaxation of the sphincter of Oddi, raising the pressure to about 20 cm of water. Because of the increase in pressure, the bile from the gallbladder enters the duodenum. The contraction of the gallbladder and relaxation of the sphincter of Oddi is regulated by the following factors. Hormonal control. The hormone cholecystokinin is the major stimulus for the gallbladder contraction and sphincter of Oddi relaxation. When chyme enters the small intestine, fat and protein digestion products directly stimulate the secretion of cholecystokinin. Neural control. Vagal stimulation also causes contraction of the gallbladder and relaxation of the sphincter of Oddi. Vagal stimulation occurs directly during the cephalic phase of digestion and indirectly via a vasovagal reflex during the gastric phase of digestion. Functions of bile salts. Absorption of fat. Bile salts are essential for the absorption of fat from the intestine. This depends on the formation of micelles by bile salts, along with lecithin and cholesterol in the intestine. Bile salts form cylindrical discs called micelles, which have amphipathic, both hydrophilic and hydrophobic domains property. Lipids are transported in micelles from the lumen to the membrane of intestinal mucosal epithelial cells, where micelles dissociates and lipids are absorbed. Emulsification of fat. Bile salts are surface tension reducing agents. Along with phospholipids and monoglycerides, they cause emulsification of fat, which is essential for digestion and absorption of fat. Source of bile acid. Bile salts are converted to bile acids in the intestine, which is then absorbed into portal blood. Thus, bile salts are important sources of bile acids that add to the bile acid pool of the body. Bile secretion. Bile salts are important diuretics. They are constituent of bile and also they stimulate bile secretion. Absorption of fat-soluble vitamins. Bile salts facilitate the absorption of fat-soluble vitamins A, D, E and K. Hence, in obstructive jaundice, features of deficiencies of fat-soluble vitamins develop. Activation of pancreatic enzymes. Bile salts activate pancreatic lipase. Prevention of gallstone formation. Bile salts along with lecithin solubilize cholesterol. Thus, they prevent the formation of stones in the gallbladder. Physiological purgatives. Bile salts act as physiological purgatives. Constipation occurs in conditions of deficiency of delivery of bile into the intestine, as occurs in obstructive jaundice. Stool color. Bile salts add a natural brownish color to the stool. Prevention of bacterial growth. Lipids and monoglycerides, they cause emulsification of fat, which is essential for digestion and absorption of fat. Source of bile acid. Bile salts are converted to bile acids in the intestine, which is then absorbed into portal blood. Thus, bile salts are important sources of bile acids that add to the bile acid pool of the body. Bile secretion. Bile salts are important diuretics. They are constituent of bile and also they stimulate bile secretion. Absorption of fat-soluble vitamins. Bile salts facilitate the absorption of fat-soluble vitamins A, D, E and K. Hence, in obstructive jaundice, features of deficiencies of fat-soluble vitamins develop. Activation of pancreatic enzymes. Bile salts activate pancreatic lipase. Prevention of gallstone formation. Bile salts along with lecithin solubilize cholesterol. Thus, they prevent the formation of stones in the gallbladder. Physiological purgatives. Bile salts act as physiological purgatives. Constipation occurs in conditions of deficiency of delivery of bile into the intestine, as occurs in obstructive jaundice. Stool color. Bile salts add a natural brownish color to the stool. Prevention of bacterial growth. Bile maintains the pH of intestinal content and prevents overgrowth of bacteria in the intestine. Laxative function. The bile salts also stimulate peristalsis, so acts as a mild laxative agent. Effect of deficiency of bile salts. Steatorrhea. Increased fat excretion in the stools, as bile salts are required for the digestion and absorption of lipids. Gallstone formation due to precipitation of cholesterol. Hemorrhage tendencies due to deficiency of vitamin K, as vitamin K fails to be absorbed in deficiency of bile salts. Vitamin K is required for synthesis of clotting factors 2, 7, 9 and 10 by the liver. Atherosclerosis. Cholesterol plaques in the walls of the blood vessel may appear. Bile salts facilitate the excretion of cholesterol into bile by keeping it in soluble state.